It's, it's about exactly what the title says, about reproducibility issues in the big data era and some statistical ideas that could be extremely useful to keep in mind. Just to start with, let me just tell you a little bit about the beginning of statistical inference, and I'm sure everybody knows about this. So at the beginning of the 20th century, essentially 20th century science followed what I want to call a Popperian paradigm, where you advance the scientific hypothesis. This hypothesis can be either falsified or corroborated by experiments, and then you design experiments to either confirm or infirm your theory. And so Popper was thinking really about the problem of induction. Like he said famously, that no matter how many instances of white swans we might have observed, this does not justify the conclusion that all swans are white. And so that's how science progressed for much of the 20th century. You form a scientific hypothesis, for example, about the usefulness of a vaccine, and then you design an experiment to test this hypothesis. Okay, so what's important in Popper's words is a scientific hypothesis it's a theory that can make predictions and whose predictions can be tested experimentally. Right, so in 1934, and then around the same time, Fisher came along. And essentially what R.A. Fisher, the founding father of modern statistics, did is he basically operationalized Popper's view at about the same time. And in 1935, he published a very influential book called The Design of Experiments, where he basically, what I just said, operationalizes Popper's point of view. And he says, well, perhaps you've seen this. You have a scientific hypothesis, and the scientific hypothesis might be that you have a lady sitting somewhere in England that declares that by tasting a cup of tea made with milk, she can discriminate whether the milk or the tea infusion was first added to the cup. We will consider the problem of designing an experiment by means of which this assertion can be tested. Right? And that has governed much of scientific research, or at least a lot of medical research in the 20th century, where you formulate a null hypothesis. Here would be that no, the lady does not have this ability, and you would give her random cups, and then you would look at the outcomes and try to decide whether or not you can reject the null. Okay, so it's been done in one concept, one paradigm set forth by Fisher is a p-value, which is a probability of observing an outcome as extreme as the one you observed under the standing null hypothesis. I assume most people are familiar with null p-values, but if that's not the case, can you please let me know? Okay, a few people, but only a few people. Okay, so the p-value is really this concept of comparing the outcome of an experiment with what you expect under the standing null hypothesis. Right, that's a p-value, and so small p-values mean, well, the outcome is not really compatible with the standing hypothesis, and large p-values mean like you cannot reject this null. You cannot reject the current theory based on the experiments. Now has something gone wrong? So if you open the popular press, and I'll just show you a few examples, but there are many, many like this nowadays. Here's the cover of The Economist, October 2013. How Science Goes Wrong. It's a full series of articles about how bad of a job we do as scientists. When you turn on and you open The Economist, you'll see scientific research has changed the world. Now it needs to change itself. And basically what The Economist is reporting is that science used to self-correct, but it does not self-correct anymore. Okay, and so there are lots of prominent scientists, as I see a train wreck looming, blah, 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 lots of cartoons with scientists putting stuff under the rugs, and so on and so forth. And so this is what is reported. And so if you go into details and into what's been reported in this article, you'll see it goes through a long list of scientific experiments that people try to replicate and could not replicate. Okay, so I'll just cite two of them, which I find particularly telling. Amgen, it's a US-based company. They took what they consider to be landmark studies in basic cancer science. We're talking about articles published in Nature, Science, all the top journals. They took 53 studies they consider to be extremely important, and they tried to set out to replicate them. And they could do this in only 6 cases out of 53. So in 47 times, somehow the effects had disappeared. Healthcare Bayer, which is a German equivalent to Amgen, they did the same. They took 67 studies, and they could replicate less than a quarter. And this is not news, really. We have this reproducibility problem. I got in contact with this thanks to Yoav Benyamini, who told me early on that if you look at the Food and Drug Administration, they have a very strict protocol on how you can advance, go through phase 1, phase 2, phase 3. By the time you reach phase 3, it should be a slam dunk. And yet 50% of trials in phase 3 end up in failure. And what's even more concerning is that this number keeps on rising with time. Okay, so I told you the press likes this stuff a lot. This is a New Yorker. The truth wears off, and it's articles that are published, littered with terms like significance chasing, publication bias, selective reporting. There's a researcher at my university, John Ioannidis, who wrote a very influential paper, a paper that he cited a lot, who explained that he believes that most published research are false, and in fact he has an estimate, which is very, very high. Okay. All right, that's the New York Times, and it keeps on going. And a personal and societal concern here is that I believe that, I've been told that trust in science is an all-time low in the US, and articles of this kind cannot be too good, right? There's a great danger in seeing erosion of public confidence in science, which is real. But it seems like the scientific community is beginning to respond and take the matter very seriously. All right, so I'll show you some initiatives that I think are noteworthy. One is called the Reproducibility Initiative. So if you're a researcher in a lab and you're worried about replicability, then there is this kind of exchange service where you can submit your protocols to this exchange service. And for a fee, obviously, they will try to replicate your findings before you go out and publish them. And sometimes they would actually select projects for free and try to replicate your findings for free. Well, that's obviously a step in the right direction. Okay, that's great. Here is the editorial of Nature that perhaps some of you have seen, April 25, 2013. Nature openly recognizes that a lot of the stuff they publish does not stand up to close scrutiny. And what they have is they completely changed their editorial line about the way you can publish papers. And so all the boring stuff that they would never show you, like statistical models, code, stuff that you get to claim what you're claiming, now they want to see. Right? So they have an 18-point checklist that you have to go through if you want to publish your work in Nature. And of course, many leading journals have similar initiatives. Science has one too. And so there are a lot of. Now, we can be cynical about this, and maybe that's dinner conversation, but it's a step in the right direction.
Is it generally considered a problem only in life sciences?
No, I don't think so. No, it's not. By and large, we're discussing biomedical sciences, but it's not only a problem in the sciences. It's across sciences. Okay, and in fact, if you want to know more about this, just to tell you how people are serious about this, in April of this year, I had the privilege to attend the meeting of the National Academy of Science, and the president of the Academy actually decided to address the Academy with exactly this issue. And his speech is online, if you will, and you'll see that he mentions a lot of problems that are outside of biomedical research. Okay, so it seems that it's getting a lot of national attention, and that's a good thing. Okay, so I think we hear about big data a lot, and so we can ask, well, what's new under the sun, right? Data gets bigger, and so what? And I'd like to think that something has changed, that 21st century science is a bit different than 20th century science in the sense that we reverse this Popperian view of science. That is, we collect data first and then we ask questions rather than formulating theories and then collecting data to validate the theories. Okay, so we have completely reversed the paradigm where we collect data first and then we ask questions. And the outcome of this is you have very large data sets available prior to formulation of scientific truth, and somehow you're going to look at all these things you could possibly discover, And it's very different from the hypothesis-driven research that we're living in so far. So what statistics has to offer, which is a second bullet in my title? Well, statistics is a science that lets you understand the reliability of what you're finding in the context of all what you've explored and lets you quantify this. Okay, so statistics has a lot to offer to this. Even though it was designed for the Popperian paradigm, it has a lot to offer in the big data world. Okay, so why is it that most discoveries may be false? So nowadays, and especially in psychology, in neuroscience, you set up a lot of number of hypotheses that you'd like to test simultaneously because you're looking at many different places. And honestly, before you do this, it's just blue sky research. How many genes have something to do with something bad? Typically not many. Yet since I have no clue, I'm going to look at them, all of them. And so we have lots of hypotheses that we wish to test. And so perhaps we're lucky, and I'm saying we're lucky, especially in the kind of studies we see now, like perhaps there are 100 potential discoveries to be made. And these are indicated by these yellow squares. Out of these 1,000 potential discoveries, there are really what I want to say 900 nulls and 100 non-nulls discoveries to be made. Now what you can do is you can go, and so let's put them in a corner. And what you can do is you can go out and test each of these hypotheses. And so if you have a study in which your power is 80%, so for those of you who don't know what is a power in a statistical analysis, it's the probability of detecting an effect when it's there. That is the probability of detecting a yellow square, which will then turn into green because it's been detected. Then we're going to discover about 80% of these 100 things we could potentially discover, right? So we're going to make about 80 discoveries. But if I test at the 5% level, then I'm going to also have 45 false discoveries. And so when I go out and report the green and the red, of course, I don't know which is which, so I report both. Well, what I can see that there's a large fraction of false discoveries. Okay, in this case, it would be 36%. And this problem gets worse if, for example, the power drops, because when the power drops, that is your ability to detect weak effects, because perhaps the sample size is small. When the power drops, then suppose now it's 30%, then you have only 30 true discoveries. The number of false discoveries has not changed. It's still 45. And so now there's far fewer greens than red, and actually most of what you end up reporting is false. Okay? All right. Now you'd say, well, 30% power is ridiculously low. Well, my colleague John Ioannidis from Stanford did what people call meta-analysis, where they look at thousands of studies simultaneously and report what the power of these studies are. And so here we're looking at an example in neuroscience. And so here you see the histogram of power in neuroscience. And you see that the median power, that is in half of the studies, the power will be lower than 20%. So you are in a case that is worse than this. Okay? Hence, a potential very, very high false discovery rate, unless you do something about it. Okay, so many, many studies are prospective with very low power, and so you're prone to suffer from the issues we're discussing at the moment. Okay. All right, so statisticians have been extremely interested in this. So, what I'm trying to say here is that there's a well-documented crisis in reproducibility in science, and of course, I cannot address. I don't have any solution, right? And I don't think anybody has solutions today. What I'm going to limit myself to is kind of present to you statistical tools so that, when used properly, may address some of these issues, right? I cannot address fraud. I cannot address incompetence. There's a certain form of selection bias that is difficult to address retrospectively. But some of these things we can possibly address, and so I'm going to discuss some of the methods to address some of these issues. Okay, so I'm not proposing to fix science. I'm proposing to put out statistical tools that will do something good. Okay, so in a very influential paper written in 1995, you have Benjamin and Josef Hochberg proposed a procedure for controlling what they call the false discovery rate, which should be very natural now, given what we've seen. Which is, well, okay, so I have a lot of hypotheses, and really now if you look at people in neuroscience, they're testing billions of things simultaneously. So I have a lot of hypotheses I wish to test. I have a testing procedure, and what you want from a statistical procedure is to control this quantity. That is, there is this quantity which is fundamental, which we're going to call the false discovery rate, which is the expected fraction of the number of false discovery over the total number of discoveries. Right? So this is a random quantity typically. You don't know what it is, but you would like of a statistical procedure to guarantee that this false discovery rate is below a target level that you could specify. So for example, we could argue that, well, it's okay, when we do science, we have to follow false leads, but we don't want to spend more than 10% of our time or our money or our postdoc time or our students' time or students' careers. We would like to have a statistical procedure that would reveal some genes, for example, so that, yeah, maybe not all genes are relevant to the phenotype I'm studying, but 90% are. Okay. And so it's a very natural type 1 error. And what's great about the Benjamini-Hochberg procedure, it comes with a procedure that if the test statistics you have for this hypothesis are independent, will actually control this criterion and will be the best in some sense, right? And it's been widely used, not as much as it should be, but it's been widely used, and it has had quite an influence in genetics, for example. So what is this procedure does? It says, well, for each hypothesis, and we may have thousands of them, I have what we discussed at the beginning, I have a p-value. And so remember, small p-values are evidence against the standing hypothesis, and large p-values are not. And so what the procedure does is something which is extremely simple. You will sort the p-values from most significant to least significant, and you're going to try to reject the hypothesis up to a cutoff. And that cutoff is very simply determined. What you do is you set your target level Q, so let's say my 10%. I'm OK with 10% of errors. All right, and then you have the line with slope 10% or slope 0.1. You plot the sorted p-values, and the last time the p-values cross this line, that's your threshold. That is, you're going to reject everything below this crossing point, and you're going to accept all of this. That is, if these were some genes, you will investigate those genes, but not these. Okay, what's remarkable about this procedure is that this procedure is adaptive. That is, if there are many more discoveries to be made, the significance level, this cutoff value, is constructed adaptively from the data. This significance, so here we can see that we have lots of p-values, very strong effects, things that I would like to reject. This significance level is changing because when I can make more discoveries, if I want to control the fraction, I can discover more. Okay. And so it's what's a beautiful property of this procedure is it's adaptive to the number of non-nulls. Okay, and so what they proved is that they proved that if you have independent p-values, then this quantity will be controlled at the 0.1 level. Okay. Okay, so of course they are not the first to study multiple comparisons problems. Some giants in the field have looked at things like this before, but I think it's a modern take on this that has enormous potential and applications. Okay, so what is false discovery rate? It's a form of what I want to call selective inference, and if I can achieve anything in this lecture is to convince you of the importance of selective inference. What the FDR is really, if you know about type 1 errors, the probability of declaring something is significant when it's not, right? It's not really a type 1 error. It's an average type 1 error, but average over the number of claims you make, not average over the total number of claims you could possibly make, right? And that's quite different. It's a type 1 error, which is average over the selected discoveries, and that's what you want when you don't want to do modern inference. You see, the way classical statistics work is you specify a model ahead of time, then you see data, then you perform inference. Nobody does this. You get the data, you look at it, you come up with a statistical model, and then you try to make inference. But that's very dangerous. All right, and so what selective inference is, it says that you want the original property of inference, of induction, or whatever you want to call it, of inference, to hold on the average over the selected. And what that means to select it, for example, for a subset of interesting stuff that you picked out after looking at the data. Everybody does this nowadays, right? So in the big data era, I get my data, I look at it, and I'm going to build a statistical model after looking at it. And that's not what the greats have taught us. And we need to adapt to this new environment, and that's the subject of selective inference. So selective inference is not just about multiple testing. It's broader than that. So for example, suppose that now there are many parameters of interest that I might be interested. So these are, think about n are very, very large. So I have very many parameters of interest, theta n. And I'm sure everybody in his or her life has studied the confidence intervals. And the confidence interval is a random interval constructed from data that will cover the true value of the parameter with some probability. And what that means is, by definition of a confidence interval, is mean that on average, if I have lots of parameters and I construct 95% confidence interval, 95% of them will cover what I want. So on average, things are right. But that's not how we use confidence intervals. Instead, what we do is we're going to select confidence intervals. So let me perhaps show you this. Right, so I have lots of paper clips like this. Chocolate makes you slim. Eating chocolate is good to get a Nobel Prize. If you eat a lot of spaghetti sauce and pizza, you will not have prostate cancer. Lots of stuff like this. And it's because people are looking at many effects simultaneously and not adjusting their conclusion for the multiplicity of the parameters or the effect they're looking at. Right? And that's something that is regrettable because it leads to this lack of reproducibility, as I've mentioned before. And that was pointed out in the same paper by Sorich in '99, which is interesting. Sorich is a medical doctor. And he said, well, in a large number of 95% confidence interval, well, we know that 95% of them contain the population parameter. That's the definition of what it means to be a confidence interval. But let's follow Sorich. But it would be wrong to imagine that the same rule also applies to a large number of 95% interesting confidence intervals, stuff that you'll select after you've looked at the data. All right, and so here's an experiment. Right, so here we've got parameters, maybe in blue, and then we have point estimates, and then we have the gray bars, which are confidence intervals, and so we have 20 things, and I draw 90% confidence intervals, and yeah, it's all right, 17 out of 20 cover their values. That's perfect. But if now I select the promising leads, the stuff that, for example, does not cover 0, telling me that there's something going on, which are these 4 guys, then they never cover their song except 1. Right? So 1 over the 4, if I select now after looking at the data, and I just report those things as if I did not select, if I did not selected them out of the sea of stuff I could have selected, then of course I don't have this property anymore. And of course these experiments will tend to fail when they're replicated. Okay, all right. So when you ignore selection, this is what the kind of stuff you get, and many things like this. Okay, all right. So ignoring multiplicity of intervals. So you see a lot of selective reporting where people have study lots of different number of food intakes. So for example, this paper where eat ketchup and eat pizza and avoid prostate cancer. It's because these researchers are looking at many types of food intakes, here 130. What you will make it to the abstract of Nature and Science are typically the 2-3 more significant findings. Typically they're unadjusted for multiplicity, right? So the abstract reports only the 3 marginal 95%. And so if you do not adjust, you end up making conclusions that are dubious. Yeah.
I mean, I thought these issues are statistically sort of well understood. So they are ignoring the statistics?
Yeah, I mean, very few people will take a multiplicity into account when reporting results. In fact, it's a very interesting homework for people to do if they're interested, is you can open a top journal and look at what's being reported and ask yourself, is what has been reported, has it been selected out of multiplicity? And very often you'll say yes. Has multiplicity been taken into account when making claims? Most of the time you'll see no.
I see.
And so this is widespread.
So it should just be, it should correspond to increasing the sample size. That's all.
No, it's not a problem of increasing the sample size.
If you do that, you'll be okay.
No, it's a problem of what you might want to think about doing Bonferroni correction. It's nothing to do with the sample size. It has to do with the winner's curse, that there is a selection bias, that the maximum of random variable in expected value is not the expected value of any of them, right? There's a selection thing, right? This is exactly what I'm trying to say here. That has nothing to do with sample size. It has to do with what you want to inference about has been selected after looking at the data. It's not a sample size issue. It's a selection issue. Okay. Please don't hesitate to interrupt me if something isn't clear. Okay. All right, so if you do not, just to show you a little bit of the paradigms that people have developed to deal with these issues in the context of confidence statements, Beniamini and Yekutieli in 2005 proposed kind of the pendant of the false discovery rate, but at the level of confidence intervals, right? So you collect data about theta 1 through theta n. I look at the data, and then I select stuff of interest. So I select a subset of parameters. And now they want the properties of inference to hold over the selected. Right? And so that leads to a new notion of false coverage rate, which is, the notion that might be, and that's, you should know that in statistics, these things are hotly debated at the moment. Is it the right notion? Is it not? Lots of discussion in the field about this, which I think makes the field very exciting because we're thinking about the selection problem and how it should be formulated, what quantities we should try to control and what not. But the proposal of Beniamini and Yekutieli is to say, well, out of those you've selected, make sure that you have 95% coverage. Not out of the whole, but out of those you selected.
Can I ask a question?
Right.
So, so in high energy physics experiment, there are issues that sound like very similar.
Very similar, yes.
Out, which
yes.
they call look L square effect.
Yes.
Where they take large number of data, and they suppose you discover a part element signature of the that can appear in any place.
Right.
And so they have a way of.
Because if you wait long enough, noise will make the signature, right?
So they have a systematic way of adjusting the number of sigma that is required.
Yeah, the 6 sigma theory or 5 sigma.
That's the same kind of issue.
Yeah, it's the same kind of issues. It's like you're looking everywhere. You're looking at many phenomena simultaneously, and you want some level of confidence about what you're finding. So in your world, it would be like I'm looking at lots of energy bands simultaneously. And so there's a multiplicity problem. And so the level, the burden of proof has to be higher. Right. No, I'm not criticizing energy physics. I'm convinced that we found the Higgs. I heard some very bizarre talks about it, but I'm convinced.
I guess the question is, you had 130 different medical teams. I guess the question is, if you had 130 medical teams, each studying one food and its effect on cancer, then the one paper that gets accepted to Nature is the outlier,
Of course.
and yet no one team behaved badly.
Yeah.
It's the field that behaved badly because they only tested one thing.
Right, that's right. But that, I think, yeah, absolutely. I mean, so that's why people do meta-analysis. That is, then they look at the outcome of several analyses simultaneously, and they try to draw other conclusions from. Okay?
So
Eva.
in these theorems, you're making the assumption that these experiments are independent? How reasonable is
Well,
this?
all what this theories require is I have honest statistics, that somehow there's a way to calculate the distribution of what I'm measuring under the null. And if that's not the case, if I don't have any way of calibrating what I'm expecting under the null, that this treatment has zero effect on my health, then Yes. Okay. So I think we have lots of topics for dinner conversation. In fact, that is one of the things that people talk a lot about. Like, perhaps one of the things that we should enforce as a nation is that every federally funded research should be reported somehow, so that even negative results can make it. So that when people are about to say that pizza is great for prostate, then maybe there are 12 in a repository somewhere. There are 12 studies that say no, pizza is no good for. And before you go out, you know. I mean, look, Mendel's law of inheritance. I'm not sure I understand what you're saying, actually. Right. Is that the behavior you want to encourage? Data fudging because I have some. No, because that's what we see, right? Sure. Is there anything wrong in applying a higher level of criticism when you're looking at many things simultaneously? Yes. Okay. I'm happy to keep on going with these conversations. All right, okay. All right, so this is something you may criticize, this criterion or not, but again, Beniamini and Yekutieli came up with a recipe that is, if you're selecting, then you have to enlarge the confidence intervals, right? You cannot just use a good old marginal confidence interval, you're going to have to make them larger. And if you make them larger, then you have some assumptions about your statistic, then this kind of very intuitive notion of what it means to do selective inference holds. That is, no less than 5% of your confidence interval over what you selected after looking at the data. No less than 5% will not cover what you want them to cover. Okay. Okay, so selective inference is that we want inference to hold when selecting promising leads. And that's what people do, that we want, people build models after data snooping. And so we want inference to hold after this has taken place. Otherwise, what kind of science are we doing? It's not classical. That's not what you see in classical textbooks. And it's worse than this, that if you look at elementary stats textbooks, they will actually select a model for you and then proceed by actually deriving confidence intervals as nothing happened. Right? And so, okay, so we're discussing a modern topic. Okay, so in the last 15 minutes, then I thought maybe I should tell you a little bit about a little piece of work we've done with my former postdoc, Rina Vogel-Barber, who is now at the University of Chicago, and just take this as it's a little contribution to this field. Okay, so we were motivated by genome-wide association studies, which are commonplace nowadays. And what you have is you have a trait, a quantitative trait, for example, like cholesterol level. And of course, you'd like to understand the genetic underpinnings of such a thing as high cholesterol level. And so you would like to understand which mutations or which locations on the genome can possibly affect a phenotype. All right, so you're trying to assay a lot of markers on the genome simultaneously, maybe 300,000, and that's extremely commonplace. So now, what do people do in this space? Well, they will try to fit some kind of linear regression model. So linear regression is still the most used model in statistics. And by the way, here, for those of you who know about high-dimensional statistics, you're in very high dimensions, so it's very difficult to move beyond linear functions anyway. And so you're going to try to posit a linear relationship between the genetics and the the genotype of a person and the phenotype. Of course, we know that it's an approximate model. We know it's not perfect. We know all of this, but we have to start somewhere. So Y would be the cholesterol level in patients. X will be genetic information that I have. So for example, the J's column of this matrix X might be the number of alleles of a certain kind that I have on the genome at that location. Right? And the goal of this model is to relate the genetic information to the cholesterol. Note that different from machine learning, I'm not trying to predict here. I'm not trying to predict cholesterol level. I want to understand which locations on the genome are truly associated with cholesterol level. Because if I want to predict cholesterol level, what do? I test myself. I don't go and genotype myself. Okay, so it's very different from what you hear in machine learning. And of course, this model is inexact, so we have lots of factors that will not account for, that are not accounted by genetics. Okay, and so people do this. People know the lasso, which is like the modern least squares of trying to do least squares, but to regularize least squares by some sort of L1 penalty that would enforce sparsity. People are familiar with this? Yes? No? No. Okay, so traditional least squares, you just minimize the residual sum square. Here you would get 0 because you have more genes than patients. And so what you do is you say, well, that's not good enough. I'm going to try to regularize it. So I'm going to try to kind of, people talked about ill-posed problems this morning. So you're going to try to minimize the residual sum of squares, the goodness of fit plus a penalty. And this penalty is the L1 norm. It's simply the sum of absolute values. And what's known about this is that beta hat, which is what we call the lasso solution, will be sparse. Like it's a method that fits a model and select variables at the same time. And so there will be many locations where this coefficient is 0 and coefficients which are not 0, presumably those locations where interesting stuff is happening. It's your decision variable. So this minimizer is minimized over beta hat. Right? So you postulate this is a classical regression model, and you don't know what is beta, so you're going to try to fit it. It would be foolish to fit it by least squares because I have more variables than observations, and so you're going to try to regularize your extraordinarily ill-posed inverse problem. Okay? And you regularize it by the lasso. Like, you could Google the lasso and see how many hits you get. Beta hat is your fit. It's your estimated coefficients. That's the answer.
X is your data.
X is, okay. Xij is a number of alleles of recessive type at location J. So it's, if I is Emmanuel and J is marker 30, like how many recessive alleles do I have at this location? 0, 1, or 2? Yes, right. And that's what people call the lasso solution, which, well, it's adding a penalty that penalizes sequences that have large L1 norms. It's trying to promote sparsity. To promote sparsity. Right? If I do L2 norm, it's basically a very linear shrinkage operation. It's like, if you might be familiar with Wiener filters, that's what you would get typically by Wiener filter. By using L1 norm, you promote enormous sparsity. Let me show you perhaps an example. So in any event, this selects a model because it fits coefficients. Most of them will be 0, and only a few will not be 0. And you would like to say that those that are not 0, well, they are true discoveries. All right, so this is the problem we're looking at. And I'm a bit worried about my time now. OK, so that's the problem we're looking at. And what we'd like to do is we'd like to do a bit what Benjamin and Hochberg wanted to do, but in a more difficult context where it's in a regression context. And I would like to find features such that when I give the biologist a list of features, the ratio between the number of false positives and the number of features that I give is bounded by, say, 10%. Okay, I would like to control the false discovery. In English, I'd like to find lots of features without many false positives, so I'm not wasting my time investigating those. Okay. So here's a synthetic example to just show you perhaps. All right, so maybe let me go back. If lambda is 0, what is the solution to this problem? Lambda is infinity, what is the solution to this problem? 0, very good. And so as lambda is in, you start at infinity at 0, and then you decrease, and then stuff will come in the model. You select more and more. Okay? So here is an example where it's a synthetic example where I have 1,500 people and 500 variables I'm interested in. And then I'm feeding this optimization problem. And as you can see, I have 500 variables, but most of the estimated coefficients are 0. And only a few, in this case, I think 46 are non-zero only. Are these 46 discoveries? Am I confident that I can go out and report? Right? So is this a false positive? Is this a true positive? And we're discussing stuff that is commonplace now in science. Like people would fit models like this, maybe even more sophisticated model, find non-zero effects. OK, you report them, and so on. Right? So is this a false positive? Is this a true positive? And how do I even begin to understand this question? Well, it turns out that because now it's a simulated example, which I know is a ground truth, well, I know actually it's a false discovery proportion, and it's very high. All these green points are true discoveries. All these black points are not. And so if I were to report these 55 guys that the lasso picked out, then there will be 26 false discoveries in there. So my false discovery rate is about 50%. Half of the stuff is not reproducible. No, I just did something extremely. I'm doing exactly what I said. I'd fix the lasso with a value of lambda which is 1.75. That's all. And that's the false discovery rate is when you have a green point, it's a point that was not, that the true coefficient is not 0, and the estimated coefficient is not 0, and the black point is the estimated coefficient is not 0, but the truth is actually 0. It's feature set. Of course. Of course. Of course. So if I were to report this, I'd be a bit in trouble. Yes. Right. And how do we, OK. So I crank up lambda, I have a different picture. All right, so basically, if you crank up lambda, I mean, these are all good questions. I don't want to take too much of your time, but these are good questions. So when you crank up lambda, what will happen? There will be fewer non-zeros. And still, I could ask the same question, like who is true and who is not true? Yeah. That does not perform variable selection anymore. Nobody is 0 anymore. Sure. Right. Look, when I tell you that there's an industry of people doing what I'm saying, I really mean it. There's an industry of people doing what I'm saying. OK. So what I would need to know is that I would need to crank lambda to estimate the false discovery proportion. But to estimate the false discovery proportion, statistically, what I need is I need to look at this guy and I need to say, let's test the null that this guy is 0. But to test the null, I would need to get the sampling distribution of this fitted coefficient. There's no way we can do this. Right? There's no way. The sampling distribution, even if I assume that this coefficient is 0, depends on all the others. And the great crime of the 21st century is that we use the lasso more than we use least squares. It's really the least squares of the 21st century, and we have no statistical sampling theory for it. Okay, so I would say, I would not know how. It's worse than that. Forget it. Okay. Okay, so in the last 5 minutes, I'll introduce an idea, which is a knockoff filter. And so knockoffs, they are just fakes. So it's not a name brand bag. It's just a cheap knockoff. Okay, and so what we're going to do in these 5 minutes is that for each variable, so now let's give a name to variables. These are SNPs. They are single nucleotide polyforms. It's my genetic makeup at a given location. So for each SNP, I'm going to construct a fake SNP. Now this fake SNP will be constructed by the computer. I don't need to measure anything about me. I don't need to look at my cholesterol level. Not anything like this. It's going to be constructed by the computer. And what this fake SNP will do is it will serve as a control group. It will it will serve to calibrate the significance of fitted coefficients. Okay, so now this construction of fake, right? So I have vectors in Rn. These are my features, my vectors. And they have some what we statisticians call a covariance matrix, right? They have correlations, and so we have two features. And when I'm asking for my fake variables, I say that the covariance, the correlation between the fakes must replicate the covariance between the true variables. Okay? But that's not only this. I need to do one more thing. I would like that the covariance, that there's a correlation between a fake and a true variable when they are distinct, is also faithful. Okay? And that's pictorially represented like this. I say, this is my fake, and I want that it correlates with this true feature just the same way that this does with this one. Okay, that's, now you'd say, how do you construct this? We have a software package to do this. You do this by matrix computations or numerical optimization. The point is that you don't need new data, and I never looked. It's a matrix problem. It's nothing to do with stats. Nothing at all. With experiment science or stats, it's just a matrix problem. You have a package that does it for you. Now the computer will speak out these fakes. Okay, now why do we construct these fakes? We construct these fakes for a very particular reason, which is that suppose SNP J has nothing to do with cholesterol. And suppose I correlate the cholesterol with this SNP. Then, because Y is a response, it's my linear model, it's the mean plus the noise, then perhaps what I should just say, and in the interest of time, I should not try to prove this, but the way these knockoffs are constructed, they're constructed in such a way, these two relations, they guarantee that if somehow variable J is not in my model, then this correlation is distributed exactly like the correlation with the fake, with the knockoff. So by looking at the marginal correlation between the response and the knockoff, it is a faithful estimate of what it is when you're not in a model but a true variable. And so what that means in terms of statistics or probability is that what I have, you see, what I'm going to have is I'm going to have all my genes over here, and I'm going to have all my fake genes over here. But when a gene is not associated with response, what I'm saying probabilistic is that these are exchangeable. They cannot be distinguished. Right? And not only they're exchangeable one by one, but they're exchangeable pairs by pairs. That is, that I can replace knockoffs and true variables, exchange them, and at the distribution level, nothing changes. The original features are completely arbitrary. It works no matter what the initial design is. It's always true. Okay. Okay, and so what we're saying mathematically is that a very simple property, I mean, it's not simple, but it's like if I have a set of genes that are not associated with a response, and I were to substitute them with their knockoffs, I were to flip them, then nothing will change in a distributional sense. The distribution is identical. It doesn't change. Okay. So I'll give you a little bit of a punchline now. What can you do now? Well, we're going to look at the examples we've seen before, where now we're not going to perform regression on the original variables, but on the original variables and their fakes. Right? It's exactly the same setup. Y hasn't changed. X hasn't changed. The Y and the X have not changed. Now I added this kind of control, in some sense, and now I perform regression, exactly like this. But now I have the variable that were the true variables, the physical stuff, and then I have the fake stuff. And I see that a lot of fake variables are selected. And that's telling me something, because they cannot be in the model. I made them up. Right? So the lasso will select 49 of this, 24 of them over here. And from this exchangeability property that I'm telling you about, well, what it says automatically is that there are probably 24 false discoveries in here, which will lead you to an estimate of FDP, which is 24 over 49, which is unbelievably close to what it used to be, 47%. Right? So what we're saying is because of this exchangeability between true null genes and fake genes, when you count the number of discoveries, it's a good proxy for the number of false discoveries you're about to make. Okay. Are we on board? Okay. All right, so now we can operationalize this whole process, and we could say, we can have a very operational procedure, which is, okay, so we're going to look at the first time a variable enters in the model. We're also going to look at the first time its fake companion enters the model. And this exchangeability property means that when I look at the first time I enter the model or the first time I fake, if I'm not in the model, who comes first is 50-50. It's a coin toss. And what you see is this. You see this statistically. You see that all the black variables, so here we see the time where the first variable enters a model. We see the time where its fake enters a model. We see the 45-degree line over here. And what this exchangeability says is that the distribution of the nulls, the things that are not in your model, should be symmetric. And that's exactly what we see. The guys that are in your models, though, should enter early, and they should enter before their control. And so they tend to be here, right? Because it means you enter before your knockoff, and you enter early. And that's what we see as well. OK, and so now you kind of operationalize this procedure where you're going to rank variables as to when they enter in your statistical model and whether they enter before they're fake or not. OK. All right, so you do this. And now with the picture, I'll show you what happens. OK, so I start fishing for explanations. And here's my first variable. It comes in, and you see a $100 sign because it's a true physical thing. I mean, it's not a knockoff. And the second is also not a knockoff. These are true genes. OK, it's a gene, but then you see a knockoff. And what it says that probably there's one false discovery in here already, I mean, statistically speaking. And so you continue, you go, and you stop. Now you have how many false discoveries proportion I want? I want 10%. So you're going to keep on adding stuff. Of course, you will never report these guys, because it's fake stuff that entered before real stuff that cannot be reported. You will only report those. But when do you stop? When you stop going on this lambda trajectory, you stop the last time the ratio between the fake and the truth is below 10%. Like that's your estimate of the false discovery rate. Right? That's the last time you stop. OK? And that's what you report. And the theorem is that, yes, that controls the FDR no matter what. Like if you follow this procedure, then no matter what the correlation is between the variables, no matter what, by the way, I've never asked you what is the noise level. You don't need to know that. That will control the false discovery. It's guaranteed. OK. All right. So this is kind of some ideas of tools you could do to control these things. What you'll see in the literature is people will try to create fake variables by permutation methods, by breaking the association between my cholesterol level, by putting Dan's genotype in front of my cholesterol level. When you do this, you create fake features, but they're not faithful. And so now the fake features are decorrelated with the truth. And so when I fit my lasso model, I look at the fake features that are obtained by permutation, and they look nothing like the nulls. Because you break the correlations, and that's a mistake. This permutation, when you do permutation method, you will be faithful to the correlation between variables, but you will break an important things, which means that this can certainly not serve as a control for this. Right, and so if you were to use permutations methods, you would absolutely lose replicability. And so maybe in the interest of time, I'm out of time, right? So maybe I'll just say that we've looked at HIV studies. Of course, this is an example where people mentioned before, you don't know what is a ground truth, but people have performed longitudinal studies, and so they have some ideas of what mutations are associated with drug resistance in some treatment. And maybe during dinner time, I can tell you that when you run our method, this method discovers a lot of mutations that happen to be reported elsewhere, and makes very few discoveries that are a total surprise to you. Right? So of course, we don't know what is a ground truth in these experiments, but when we run the method, it behaves extremely reasonably, okay? Hinting at strong reproducibility. Okay, so these are HIV data that my colleague Schaefer from Stanford gave us. It's a wonderfully curated data set, and when we try this method on this data set, then we can find an enormous agreement with what people have independently thought were important mutations associated with drug resistance. Right, so I don't want to take too much of your time. I'll just finish this since there's this important field of selective inference, which is what do you do when you select a statistical model after viewing the data? It's a critical issue, I think. Statisticians are extraordinary engineers. There's a lot of activity. And I think it's a fun field because it's a fun field for the reason that we don't know really what are the right concepts. And it's good to kind of develop the right concepts. Thank you very much.